Down to here, second to last show for the semester here. So we're going to be talking a little bit about now the sense of vision, and really this first one here is going to be a lot about talking about the different parts of the eye. So really the anatomy of the eye. So outside of the eyes, we're also going to have a number of accessory organs. So the eyelids, the different extrinsic eye muscles. So those would be the ones that actually control the movement of the eye, not the ones that are on the interior of the eye, as well as your tear ducts and your tear making materials there. So kind of in contact with the eye and in their lining of a lot of the parts of the eye is what is called the conjunctiva. If you've ever heard of conjunctivitis, like pink eye, that is infection of that with a bacteria. So you have this ocular conjunctiva that goes over parts of the interior eye as well as the inside of the eyelid, which is that palpebral conjunctiva, and then you have the fornix where they connect to one another. Lots of goblet cells in here, lots of kind of mucus to moisten the eye, lots of blood vessels, quite vascular with the exception of parts of the actual eye that do not have a lot of blood vessels, but at least the surrounding tissues that cover the eye is very vascular. Lots of nerve endings. Pretty much all these blood vessels and everything else like that, though, do not overreach into the cornea. That lets light pass through it, so because of that, there's not going to be any blood vessels or other materials that are going to be found within that cornea. So with vision, we are able to sense a large amount of information from this. So like it says there, 10 million gradations of light, 7 million different shades of color, assuming you have normal color vision. That's how we bring in most of our information. If you start talking about all these different sensory receptors that we've discussed, the eye contains 70% of all sensory receptors. So between your two retinas, that's 70 out of 100 of receptors would be found in the eye. And so, I mean, that's kind of the ear, touch, everything else like that is that other 30%. And about 1/3 of all sensory neurons are in the optic nerve. So it's a very receptor dense key area when it comes to doing that. And then we have a whole brain region devoted to just the vision senses. We see about a small chunk of that electromagnetic spectrum, which is going from ultraviolet all the way up through that. Since we are the ones that put terms to all this stuff, we call it the visible light spectrum. It's visible to us, not necessarily to all species. There are certain species that have ability to see types, wavelengths of light above and below these numbers. When we start looking at some of the different structures, though, one of the things we'll see is these accessory structures. You have the lacrimal apparatus. This is what is going to produce, collect, and drain lacrimal fluid or tears from the eye. It keeps the surface of the eye moist, reduces friction between that and the eye sliding over the top of it, helps to keep it clean, and with a lot of substances that are found in this lacrimal fluid, generally tries to keep infection from being an issue on the eye. So there is some antibodies in the stuff. There is enzymes like lysozyme that help reduce bacterial populations that would be present on the eye. The lacrimal gland is in the upper portions of the eyes over here. It's continuously doing lacrimal fluid. That's part of the reason that we blink a decent amount of times throughout the day. This helps move this fluid onto the eyes, and then we're going to see it's going to go to the center here, to this lacrimal caruncle that is going to be responsible for draining into all the lacrimal ducts that are going to be draining this down to the nasal cavity. So you have two little openings in here called lacrimal puncta that the fluid is going to drain into, and then they go into what's called the nasolacrimal duct, which is taking this fluid from that lacrimal sac in that area down into the nasal cavity where it mixes with mucus to help moisturize the air that we're breathing in. But it's one of the ways we get rid of this fluid. This is also part of the reason a lot of times if you get upset and you're crying, you tend to have a stuffy or runny nose because a lot of that fluid would be actually getting collected down into the nasal cavity. So this is showing you that lacrimal gland up here, and this is where you have those puncta and that lacrimal sac, and then the drainage that goes down into the nasal cavity. I'm not going to ask you to trace a tear or anything else like that with these, but this is just kind of giving you that basic idea of these structures. Outside of that, we're going to talk about the different layers of the eyeball. When they really talk about the eye, they break it down into 3 main layers. You have the fibrous layer, the vascular layer, which you can see is composed of a few other things there, as well as the nervous layer. We're going to work through each of these and talk about the structures that make up each. So the fibrous tunic is the outer portion of the eye. There is a lot of dense regular connective tissue in this one. This is one of the things when we actually are able to do stuff in person. We do an eyeball dissection. You're going to be doing a digital one with the lab stuff and connect on this one. But we do normally an actual eyeball dissection. And with this one, we're going to see that the eyeball is incredibly tough. It's one of those things that people are always amazed at how much pressure you have to put on the eyeball to be able to poke a pair of scissors in there to cut open the eyeball. So you have the white part of the eye that makes up really it's everything of the eye that's not the cornea. But that is the sclera on the outer part here. It provides the shape of the eye, protects the eye, is the attachment site for all those muscles to attach. It's continuous with the dura mater where the optic nerve is coming out. The other portion of the fibrous tunic is the clear portion, that convex structure on the front of the eye, which is the cornea. This is going to let light pass through it as one of the ways we help to focus the light into the retina to have vision. So you can see the sclera is this whole part back here, where the cornea is just this portion in the front where light passes through it. Again, the cornea, because light is passing through it, we have no blood vessels, we have clear cells that are making up this. Like I said, the shape of it helps to refract light into the lens, which is going to help focus that onto this. And again, it has epithelium on each side that is going to get nutrients either from the lacrimal fluid or from fluids in the anterior chamber. The middle layer of the eye is what we refer to as the vascular tunic. Not surprisingly, lots of blood vessels here, but we're going to see that there is 3 main regions to this that are involved. So in the back part of the eye, you have the choroid, and then the ciliary body and the iris are going to be part of that junction between the anterior portion of the eye and the posterior portion of the eye. So we're going to have these intrinsic muscles of the eye, the iris, and the ciliary muscle. So you can see the iris is part of this tunic. Ciliary body is surrounding that lens, and the choroid coat is one of the layers underneath the retina in the back of the eye. So the choroid, again, the biggest portion of this has a lot of pigment in it from melanocytes. If we were actually looking at an eye, you'll see that this portion, a lot of it is black. It really limits any light coming in from any other region besides through the cornea and through the lens. Absorbs any of this extraneous light that could come through the skin and through the side of the skull, let's say. Means that only the light coming through the pupils is what's getting into the eye. If we go to the front of this, and what is going to attach to the lens is the ciliary body. So this is to the front of the choroid. It's kind of at the junction between the posterior cavity of the eye and the anterior portion of the eye. There's going to be this circular muscle called the ciliary muscle, and these little kind of high surface areas called ciliary processes. Within that ciliary processes, we're going to see that there is certain areas that are making some of the what is called the aqueous humor that's going to fill up the anterior chamber of the eye is made by the ciliary process. The ciliary muscle actually attaches to the lens by these little ligaments. And if this muscle is relaxed, it gets wider like this. It pulls on the lens and flattens the lens out. When the muscle contracts, it lets the lens become more circular. Like I said, the ciliary processes have a lot of these capillaries that are making that filtrate that is going to make the fluid that fills the anterior portion of the eye. So you can kind of see the ciliary body here. Like I said, all this space up underneath the cornea here, the ciliary processes back here are making the fluid that is going to fill this area in front of the lens. The iris, or really when you look at somebody's eyes, that's really what you pay the most attention to. That's the colored portion of the eye. You have these different pigment forming cells. Each person, it's pretty unique in terms of what's going on there. But they have these layers of pigment forming cells that can give you that wide range of colors on the eye. But really what the iris is doing is it is, in a way, a shutter. If you've ever worked with a camera, you know that too much light onto film overexposes it. Not enough light, you don't get a good image either. So you need the right amount of light getting to the camera for a good picture, it's really the same thing with our eyes. You need the right amount of light getting to the eyes. Too much light on the retina is going to lead to a bad image. Not enough light on the retina is going to lead to a bad image. This iris can get bigger or smaller to make that pupil bigger or smaller, which will let more or less light in to maintain the correct light intensity we need for making a good visual image. And you can see the picture of the iris right here. So a lot of people think of the pupil as being a black dot. In reality, it's an opening. The pupil is mainly black because light generally goes into the eyeball and doesn't leave. In absence of light being reflected back is going to be black. If you've ever seen a camera where you get red eye, that is that flash, the light going in there, hitting all the blood in the blood vessels in the back of the eye and then reflecting back out. But the iris, again, is controlling the light intensity getting into the retina to our detector. And like I said, it's really controlling the size of that pupil. There's going to be actually two different layers of muscles. There's one muscle that will dilate the eyes, and then there's another one that does constriction. And again, only one of these is working at a particular time, but that's what allows us to either open or close that pupil a little bit more to let more or less light in. We're going to see in AP2, a lot of this is coordinated by that autonomic nervous system. So you have what is called the sphincter pupillae muscle. This is a circular muscle on the inner portion of the pupil. And when this contracts, a circle muscle, when it contracts, is going to make the opening smaller. That constricts the pupil, controlled by the parasympathetic nervous system. The dilator pupillary muscle, these are arranged in like rays going out from the center or something like this. So when those contract, it's going to open up that pupil a little bit more. These are controlled by the sympathetic. And again, when light starts getting lower, these will be activated to try to let more light in. So you can kind of see it right here. Here's that circular muscle right here, that sphincter pupillae muscle. And you can see all these radial muscles right there. When those contract, it will open this up a little bit more. So again, if you're ever really bored, take a flashlight and a mirror, and if you shine the light on your eyes, you will see that pupil constrict. Or if you're in a dark room and then turn the light on, you'll see the pupil going from very dilated to more constricted. This is to control that light intensity getting back to the retina. So again, kind of our first portion here of the eye, what we'll do is we'll work